Hi again, everybody. We are going to be talking about proteins in section 5 of chapter 3. As you can see here from the slide, proteins are pretty much everything. Their defense, their storage, transport, communication, movement, and structural support. As ever, I think a picture is way better than words. And this is a real great picture of how they function inside of our bodies. They're enzymes, and the next chapter is entirely on enzymes, so we don't have to spend a lot of time talking about that. They're defensive. They're part of our immune system in the form of antibodies. They can be storage, for example, inside this eggshell, or whenever you eat an egg, you're eating a huge amount of protein in the albumin. They can be transport proteins in the form of allowing things to enter and exit the cell, or they can be transport, like hemoglobin is a transport molecule for oxygen. They can be our communicators, hormones. They can be our neurotransmitters or other signaling molecules. They can also be the receptors. They literally make up our muscles. Every movement, every time my lips move, my diaphragm moves, these are all muscle contractions that are made up of proteins. And lastly, your body, your skin, your hair, your organs, your blood vessels are all made out of protein. So proteins are really, really important as a biological molecule. Once again, we're going to talk about enzymes in a whole chapter, but they are catalysts which speed up chemical reactions. So now I want you to look at this slide. I've got some yarn here. Maybe you have someone in your family that knits, and I've got this great piece of yarn, and I want to ask you, what do you think of my sweater? Is it pretty? I know you think I've lost my mind. You're like, Ms. Corcoran, that is not a sweater. That is just yarn. The reason I'm showing you this is because I want you to understand the concept that a polypeptide is not a protein. A polypeptide is many amino acids linked together by a peptide bond. It is like a polysaccharide, many sugars linked together by glycosidic linkage. It is just many in a straight line. It is not a functional protein, just like this is not a sweater, until it's folded into the appropriate way such that it can do its job. So I hope you understand that protein folding is profoundly important. Here's your structure of an amino acid. You should notice right away a carboxyl group on one end and an amino group on the other end. Okay. Every amino acid has a central carbon. I like to say it has a hydrogen top hat, and then below it there's an R group, which is a variable group, also called a side chain. That's what gives every amino acid a different property. The next slide I'm going to show you has 20 amino acids. Do you have to know them all? No way. What you need to understand, and it's really hard to see, let me see if I can make it bigger, is this is your amino group. This is the carboxyl group. It has already donated the hydrogen, so that's the acid part of amino acid. Here's your top hat. Here's your central carbon. This is your side chain. All of these side chains are, I think, non-polar side chain hydrophilic. What is this? Hydrophilic. So these are polar. Up top, gosh darn it, I'm sorry. My bad. I don't know how to drive when it's, here we go. Up top, we have polar side chains. We've got non-polar side chains. We have acidic and we have basic. So my question to you is, do you think a polar side chain is going to want to spend any time around a non-polar side chain? No way. That's where we get into the folding of the protein. If you have a polar and a polar, they're going to want to be together. They're not going to want to be non-polar, near non-polar side chains. So that's going to really influence the shape of the protein. Oh gosh, I need to go back down to my small size. Okay. So the bond that forms between amino acids are peptide bonds. And I wonder if I have a piece of paper I can use. Remember that when we talked about two groups join, what has to happen? That we lose a water molecule. NH. Okay. Actually, just ignore what I messed it up. So when two amino acids get together, look how they're lined up. This one has already formed a peptide bond. Take a look right here. It's really important. That's your carboxyl group, and that's your amino group. If we had two amino groups together, there would be no oxygen present to help create that molecule of water. But because we have the amino group and the carboxyl group, we can easily squeeze out a molecule of water through dehydration synthesis and form the peptide bond. The reverse would happen if we were trying to break the bond and break down this molecule through hydrolysis. We would add a molecule of water. The OH would go to the carboxyl group and the other hydrogen would go onto the amino group. So I cannot stress enough that you absolutely need to know the orientation of amino acids when bonds are formed. Okay, the carboxyl group of one and the amino group of the other are going to get together. Okay, let's skip that. Shape really dictates function. Here's an example right here. You've got a protein from the flu virus, and you have an antibody that actually matches perfectly once your body has been exposed and has been able to create those antibodies. So shape is really everything. Notice how this has some gold stars and some red letters. I'm telling you right now, 9 points of your next test are going to come from protein structure and any molecules associated with helping it fold and all of the bonds. So let's go through those right now. The primary level of protein structure is simply an order of amino acids dictated by DNA held together by peptide bonds. That's the equivalent of my yarn here. It's a polypeptide. It's not a protein. It is the primary sequence of amino acids. It's not bent or folded in any specific way. It's just amino acid, amino acid, amino acid, amino acid, amino acid, linked by peptide bonds. Your primary structure. The secondary structure is, and if we were in class together, I would have given you a pipe cleaner and asked you to take your writing implement and wrap your pipe cleaner around your pen or pencil, okay, for about halfway. And you've got a coil. You see the coil? This is going to represent the alpha helix. That is the one structure in the secondary structure. The other possible structure is something called a pleated sheet, a beta pleated sheet. So we're going to bend these up and down like an accordion. So you see my up and downs are kind of not perfect, but you kind of get it. My up and down bends and my coils. So I have an alpha helix and a beta pleated sheet. This is a secondary structure of a protein. You see that it's no longer just a straight line anymore. It's no longer just a straight line. It has been bent, and this is due to hydrogen bonds. Okay, so secondary is alpha helix, right up here, and beta pleated sheet. You need to know both of those, and they're held together by hydrogen bonds. Now your tertiary level, FYI, this slide's really kind of interesting, that spider silk, the radiating strands that help maintain the web, have pleated sheets. So these proteins that make up spider web are actually secondary levels of protein structure. They don't have to have all the levels. They have to go beyond the primary level. Now your tertiary level is your 3D globular form. So I've got my nice alpha helix and beta pleated sheet, and I'm going to just gently roll it together, not trying to compress it too much, kind of put it in a little ball. It doesn't really look good. That one's a little bit better. Okay, so it's sort of a more globular 3D structure than it was before. This happens because our side chains interact with each other. Remember, we talked about hydrophobic wants to be with hydrophobic, and ionic would like to be near another charged one, and hydrophilic ones all want to be together. So this is what is the result of interaction among the side chains. You need to know all 5 bonds. Let me go to this slide. Okay, so the side group interactions are van der Waals interactions, hydrophobic interactions, ionic bonds, disulfide bridges, and hydrogen bonds. That's 5 different kind of bonds you need to know for your FRQ. That your tertiary level is your globular 3D structure of your protein due to interactions among the side chains and held together by hydrophobic interactions, van der Waals interactions, ionic bonds, disulfide bridges, and hydrogen bonds. All 5 bonds need to be listed. Okay. A quaternary level of protein structure results from 2 or more polypeptides. So I've got 2 or more structures here, and they are going to associate with each other and be the quaternary structure. So that I have 2 here, and I'm going to go ahead and add a third and just kind of roll them together. And you can see here that this is even a different shape. This would be a quaternary structure of protein folding. This is when 2 or more polypeptide chains associate together. The same bonds, and you don't have to list them again in the FRQ, but the same bonds would be responsible for holding these polypeptides together that we see in the tertiary level, which are van der Waals interactions, hydrophobic interactions, ionic bonds, hydrogen bonds, and disulfide bridges. Hemoglobin is an example of a protein that has a quaternary level of protein folding, that it has 4 subgroups. Collagen has 3 subgroups here. So not every protein will have a quaternary level. Know that it's certainly possible. Now, how do we make sure this protein gets folded? Because remember, shape is everything. Shape dictates function. So we have to make sure that the protein is properly folded. And there's a molecule in our cell called a chaperonin or a chaperone. It literally chaperones proteins folding properly. It provides an environment within the cell where they can do their folding in sort of an unobstructed or undistracted environment. So it goes in as a polypeptide and it comes out appropriately folded. Now, later you're going to watch this TED Talk and answer some questions, but it's really interesting in the research that has to do with the chaperones and what happens when poorly folded proteins accumulate in our body. So let's talk about sickle cell disease and mutations. We know that it is the genetic code that codes for the order of amino acids. Let's say that our amino acid called for a hydrophobic amino acid. Our DNA called for hydrophobic amino acid, but we have a mutation and it brings hydrophilic amino acid instead. Certainly, it's going to change the shape. And in the case of sickle cell disease, it actually changes the shape of the protein hemoglobin, which as a result changes the shape of the red blood cell and causes it to sickle. So you can see how incredibly important shape is to function. Now, here's a term you probably learned in Bio 1, and the term is denature. Loss of shape is denature. We denatured enzymes when we heated them up. A heated enzyme doesn't work anymore. Why? Because we actually broke the bonds. You know, the heat increases atoms' movement, and we broke the bonds and we unraveled our protein. Remember how this looked a few minutes ago? It doesn't look the same, and it's certainly not going to do its job now. So a denatured protein is biologically inactive. On the upside, the good news is denaturing changes the shape, but it can actually refold and renature so that it can be functional again. So you're wondering, I'm sure, how scientists know what the shape of proteins are, and they use something called X-ray crystallography, which was actually used to determine the structure of DNA. It's been around a long time, but that's how scientists know the shape of proteins. If you have any questions about proteins, just let me know. Otherwise, have a great rest of your day.